Hey everyone, and welcome back to the Ontario Aquaculture Research Centre. I'm your host, Kaylee Moore, one of the agricultural assistants here at OARC. If you're looking to learn more about aquaculture, you're in the right place. On today's episode, we have Beth Lenentine joining us from the Coldbrook Biodiversity Facility to talk about their efforts in maintaining the inner bay of Fundy Atlantic salmon. Hi guys, and welcome back to our channel. I'm so excited to introduce Beth Lenentine, the manager of the Coldbrook Biodiversity Facility. Today we'll be discussing the importance of their work in the Maritimes Region. So Beth, in the Maritimes Region, there are two biodiversity facilities. One is in the New Brunswick and one is in Nova Scotia. At the Coldbrook facility, the primary role is to kind of maintain the genetic diversity of the inner bay of Fundy Atlantic salmon. Can you give us a little bit of the facility's history?
Sure. Coldbrook Biodiversity Facility was formerly known as the Coldbrook Fish Culture Station, and it's been operational since 1938. And even back then, the focus was strictly on enhancement and stocking wild Atlantic salmon in Nova Scotia. So the facility is operated by the Federal Department of Fisheries and Oceans, and prior to us doing live gene banking, when we were still enhancement, at one point in time there were 13 hatcheries in the Maritimes Region. Over the years, some of the facilities had closed. There had been a divestiture of our facilities in 1998, and then DFO took our facilities back under their fold in 2000, and that was at the inception of our live gene bank program.
Oh, amazing.
Coldbrook is really aptly named. So from a fish culture perspective, we have a lot of groundwater influence, and our water temperature regime and access to a significant amount of groundwater certainly makes it an ideal facility for the live gene bank program.
That's amazing. Yeah. Back then, DFO's Maritimes Region hatcheries were primarily growing Atlantic salmon for stocking rivers to increase numbers, yet the population continued to kind of decline. Now you're focused on maintaining live gene bank, which you have mentioned, and how has that kind of changed the approach?
Right. And so I've been involved in fish culture for 30 odd years. And so I can definitely say that the approach of our live gene bank program, although it is, fish culture and animal husbandry is animal husbandry, a lot of the traditional fish culture practices, I guess I liken it to, the fish culture practices, definitely taking, a backseat to the processes and the programs and the activities that we did, as enhancement. So as part of the live gene bank, and I know we'll be getting into that later, is instead of releasing large numbers of fish for stocking, a lot of the background is in bringing in wild stock into the facility. So even back in 2000 when we started this is totally converting to hand feeding, not grading, because we certainly recognize that. Same as with humans, we have, people that are 5 feet tall and people that are 6 feet tall. And by grading out the 5 foot tall people, or very similar, if you use that approach with fish, you're missing a lot of genetics. And so things like, focusing on, the genetics, and we have noticed that every stock that we work with is different in terms of their growth, many different, aspects, their phenotype, many differences. So we're really focusing, we had to totally rejig the program and our fish culture practices to that specific, to a specific live gene bank approach. And so we would be bringing in, juveniles, adults, and intercepting post-spawn kelps, and bringing those into the facility. A lot of that stuff hadn't been done to the degree, that we're doing it now. And so it really was, it was certainly a learning approach for our staff as well, from our traditional fish culture practices.
No, I'm for sure. Like you're talking about, grading and stuff like that. Like that's something we don't do as well, like for our brood stock and stuff like that, because normally if you may be grading, you might be grading the one same family up and leaving the family that might be a little bit slower growth or things like that. I get to kind of maintain the brood stock part of this facility, and same thing. It's nice because I don't really have to do lots of grading.
Yes.
But like you definitely like each tank is so very different, and you can see within the population. We have rainbow trout that I'm talking about here. It's just, yeah, like every fish is almost very different. Like you don't have a very unison pairing. But yeah, because, you know, just like humans, everyone's a bit different, and genetics are different for every fish. So,
yeah.
Can you explain to us what a live gene bank is and kind of the importance of that?
Sure. You know, I guess I should, you know, first off mention that we we have a genetics unit. So we're based out of the Bedford Institute of Oceanography in Dartmouth. And as part of our DFO science salmon team, our whole program is under the direction of a genetics unit. So the genetics unit, you know, designs, you know, our mating plans for us. But I guess, you know, I'll just kind of back up a bit. So, you know, the Live Gene Bank program in the context of what we're doing is done to maintain the genetic diversity of a particular stock that we might be working on. And that might include, you know, producing broodstock or adults, you know, or bringing in or rearing through to adulthood, and doing a prescribed mating cross of those broodstock. And we're releasing the offspring from them at the earliest possible stage. So we are releasing predominantly the majority of our offspring as unfed fry. And, you know, as well, you know, maintaining the genetic diversity, you know, the early releases, you know, maximizes natural selection. So as part of that, when we release at our earliest stages, we have a very important component for our Live Gene Bank program, and it is by equalizing our family groups. So we want to make sure that, you know, in a particular area where we're putting our unfed fry above waterfalls, for instance, so there isn't any outside influence. So we are equalizing all of our families. So every family has an opportunity, you know, very same opportunity of survival. So everybody is equalized and released into an important area. And then we go back in 18 months and collect those, or, you know, up to 600 of those individuals and bring those guys back into captivity. So everyone, you know, all those fish have gone through, you know, natural selection, survival of the fittest. And, you know, so those are extremely important to, you know, continue and to maintain the whole Live Gene Bank program. So when we bring them back to the facility, like I've mentioned, you know, and that was a learning curve as well, is, you know, when we're bringing in wild fish, how do you transition them over to a dry diet? Because obviously, you know, a dry diet is totally foreign to them. And it had taken us a few years to, you know, develop our approach. That we are, you know, 99% successful with every, you know, with each group that we do bring in. And rear them through, like I said, to maturity. Every individual is pit tagged and tissue sampled. And from there, like I say, our genetics unit is able to, you know, design mating programs and yeah, for the whole Live Gene Bank program.
That's really fascinating. It's that's like a really big project, and it takes a lot of aspects to come together to, you know, have what you guys have. And part of that is research. Research is a big aspect in identifying many limiting factors for population productivity.
Right. Yep.
What is kind of like the current focus or goal to really maintain that inner Bay of Fundy Atlantic salmon?
Right. So at our facility, we do you know, a lot of trait monitoring at every life stage. So for instance, you know, during spawning, you know, every individual that, like, certainly within our inner Bay of Fundy stocks, you know, the tag number is, you know, in the image. We're doing digital images of the fish. We are collecting eggs and looking at egg diameter, egg weights, and just to look at any effects or any changes over time, digital images of all the eggs, like I say, equalizing. And so, you know, looking at, you know, from all of the data that we are collecting for our trait monitoring, we can get into things like, you know, any maternal effects, any potential for domestication selection over time, So we really have a huge number of set points, and we are collecting a lot of data just to be able to make sure that, the whole live gene bank program is successful.
No, that's perfect.
Yeah, and we also, certainly within our DFO team, within our Marine Threats Unit, and we work very closely with our assessment unit. And we're doing things like, being able to track our fry and look at, survival from, by tributary, by river, and looking at, which would then, of course, get into, developing a potential release strategy for each river that we're working on. And then we also work with a lot of academic institutions with, master's students and PhD students and tagging and tracking studies with various universities too. So yeah, so we are, pretty interconnected.
Yeah, it sounds like it's quite the interesting facility. There's a lot of different dynamics and aspects that really creates the business, not the business, but the biodiversity facility that you guys have going there. And that's all the questions I really had planned for today. I don't know if you have anything else you would love to add or tell us about.
Yeah. Sure. I mean, we do have, our main focus. So in 2000, Kasiwik listed Inner Fundy salmon as endangered. And so we do have at our facility, we have 2 stocks of Inner Fundy, Stewiacke River and the Gasboro River. But we're also branching out and doing some Southern Uplands populations as well. And, we were kind of at the cusp of, the huge decline in the Inner Fundy and, trying to circumvent the same thing happening on, with the Southern Uplands populations. So the program is probably not as robust as the Southern Uplands because, like I say, we've been doing the Inner Fundy. We have been doing the Inner Fundy stocks for, 20 years now. So, yeah, so it's very interesting.
Yeah, I'm excited. I was mentioning this earlier to you, but like getting to talk to Tim over at Fort Folly and the habitat as rehabilitation part, and that's what I think is so cool about aquaculture. And we are a research facility, so I mean we have more focus on research, but we do a lot of just rainbow trout, and so we get to see some diversity in species. But aquaculture is not just like what we're doing, but what you guys get to do. You have a stock and you're collecting the genetics and stuff like that, but you're part of restocking efforts, and kind of how everything kind of ties together and working together is just using and taking advantage of both, natural environment and also being able to use an on-land system that will benefit the wild stock, which is fantastic. And I just want to thank you, Beth, for joining me today and sharing with us the great work that you guys are doing over at Coldbrook Biodiversity, and the efforts to maintain this genetic diversity of the Inner Fundy Atlantic salmon and positive effects I know is having. So
excellent.
Thank you.
Awesome. Thank you so much.
Hey guys, I hope you liked the video today. If you did, please swim on over and hit the like button and subscribe. Comment below if you have any questions you want answered in any of our future videos. Hope to see you next time.